Good morning, students. Today we are going to discuss applications of Rdna technology. As we know that the Rdna recombinant DNA technology, in which the foreign DNA is introduced into certain host, whereby that DNA produce a novel protein or novel characteristics. So using this property of the technology, there are huge applications are listed. The scope is here about on this technology. We will see the application of Rdna technology in plant and agriculture field, medical and pharmaceutical, in environment science, genomics, diagnostic of genetic disease and disorders, in gene therapy, in industries for development of transgenic animals, and we also will see the harmful product, negative features of the organism. So we see first the application of the Rdna technology in the field of agriculture and plant. In agriculture and plant, the crop are challenged by variety of situation. So to overcome this situation, it is possible to construct certain genes and design the gene which able to plant to grow under adverse condition. So that's why we can design the plant which grow under stressed environment, heavy acid, acidic soil, alkaline soil, under drought condition. So we see one by one. The first objective is development of plant resistant to environmental stresses. We know that in India there are various climatic zones, not only in India, throughout world. So there are certain regions which are arid region. There is a low amount of the water for the cultivation. So there should be need the plant which grow under low water conditions. And there are naturally plant which have the adaptation to grow under low water condition. So the genes are identified from such plants and transported into the another plant. So that plant can become the drought resistant variety. तो आप वैसे उस प्लांट को कल्टीवेट कर सकेंगे जहाँ पानी कम होता है, और नेचुरली जहाँ पानी कम होता है वहाँ पर भी ऐसे प्लांट होते। तो उसमें से आपको आइडेंटिफाई करना है कि कौन सा जीन रिस्पॉन्सिबल है, और वो जीन ऐड करने पर जो उसकी अडेप्टेशन स्ट्रेटजी है वो चेंज हो जाएगी और प्लांट कम पानी में भी ग्रो होने लगेगा. So drought resistance variety can be cultivated. Another example of environmental stress resistant variety is a cry resistant variety. Cry resistant variety is the plant which normally not sustained at the very low temperature. बहुत कम टेम्परेचर होने पर क्या होता है, the frost formation takes place and the ice crystals break down the cell. तो पूरा जो क्रॉप है वो एक साथ टूट जाएगा, गिर जाएगा खेत में. So to sustain this one, naturally the plant which grow in a cool area climate, they have the certain genes. तो जो प्लांट ऑलरेडी बर्फ वाले जो एरिया है वहाँ पे ग्रोथ है, उसके अंदर एक प्रोटीन होता है, दैट इज अ क्राई प्रोटीन एंड क्राई जीन. So this cry gene is identified, which is make the adaptable, which gives the adaptability to the plant. So this cryprotein is transferred into the another plants. So now the another plants become the frost resistant variety. So this way the frost resistance is achieved in the plant. Similarly, the plant can be modified by finding the genes which make them to sustain under alkaline soil, acidic soil, heavy salt soil. So this is one of the achievement which is nowadays possible to find out the gene and develop the plant which can grow under highly acidic or alkaline soil. Herbicide resistant plant. In crop, in the field or in farming, herb is an extragenous grass and plants which grow along with the main crop, and they utilize the same resources which we make for the main crop. So it is common practice to remove this extra herb. But in a large field, it is difficult to remove this herb manually. जो ज़्यादा गैस है उसके साथ उसको हमको निकालना बहुत मेहनत हो जाएगी. So there is a practice to add the herbicide chemicals. Herbicides are those chemicals which remove all the herbs. जो जितना आजू बाजू में ज़्यादा घास है या अनवांटेड प्लांट्स है उसको हम केमिकल से रिमूव करेंगे. बट द सेम केमिकल ऑल्सो अफेक्ट द मेन क्रॉप. द मेन क्रॉप इज अफेक्टेड. सो इन ऑर्डर टू सस्टेन द मेन क्रॉप को आपको कुछ नहीं होना चाहिए और सिर्फ वीड्स जो भी है एक्स्ट्रा हर्ब्स है वो निकल जाने चाहिए. तो उसके लिए आपको हर्बिसाइड रेजिस्टेंस प्लांट डेवलप करना पड़ेगा. सो इट इज़ पॉसिबल. इन द फिगर यू कैन सी देयर आर सर्टेन एग्जांपल्स ऑफ हर्बिसाइड रेजिस्टेंट प्लांट which are not affected by the chemical herbicide. So glyphosate, it's the herbicide chemical which normally kill the main crop also. But now by development of the herbicide resistant plant, it is not possible to damage that one. So this way you can develop the herbicide resistant plant. Insect resistant plant. So insect are the one of the large group of the pest. Pest means the organism which damage the cultivable crop, fruits and vegetable. So when they attack on a food, fruit and vegetables on farm, they largely damage the crop, and this way they decrease the total productivity of a that decrease the total productivity or decrease the yield of the farm. So the scientists have find out the insect which die due to some disease. They find out which organism is responsible for causing the disease. What is the mechanism of causing the disease? तो जो भी इंसेक्ट पेस्ट है वो कैसे मर जाता है, तो उसमें से माइक्रो ऑर्गेनिजम्स जो थे उनको पैथोजन वो आइडेंटिफाई किए. पैथोजन उसको कैसे किल करते, देन दे आइडेंटिफाई द मोड ऑफ एक्शन. मोड ऑफ एक्शन में से पता चलेगा देयर इज अ बीटी टॉक्सिन इज देयर. सो द बैसिलस thuringiensis जिसको हम पहचानते हैं दैट प्रोड्यूस वन प्रोटीन during the spore formation, that is parasporal body, and that toxin is delta toxin, and for delta toxin they identify the gene. So this gene is identified and isolated, and then after that same gene is introduced into the plant. It is merged with the plant. So now the plant have the gene in each and every cell of the plant, and that gene produce the delta toxins. So whenever any insect fed on their same plant, that will be died out. So this is due to the presence and production of the foreign gene and foreign protein into the plant. This way, we can construct the transgenic plant. So transgenic plant become the insect resistance. So the best example of this one is the BT cotton, BT brinjal. All the plants start with the BT. Plants are also susceptible to the fungal, bacterial and viral diseases, and the farmers normally treat with the pesticides and insecticides and herbicides and certain chemicals. So to reduce the effect of this harmful chemical, it is possible to modify the plant which become resistance to the virus, fungal and bacterial infection. So this is the application of Rdna technology, whereby you find out certain genes and introduce into the plant. So this plant become resistance to the virus, fungi and bacterial infection. The mode of action and mechanism for different resistance may vary. In the photograph you can see in the center the resistance variety, which is the more greener and higher as compared to the susceptible variety on the both side. So this can show that the Rdna technology can develop the resistance variety, resistance to the disease. Plant and agriculture field. Here, apart from this resistance and stressful environment, the another objective of the Rdna technology in the agriculture field is to develop the plant which give the maximum and higher crop value, higher nutritional value, quality product and fasten the product. So here in the simple diagram you can see the same plant and the same environmental condition have a different growth level. This growth level may be varied by changing the respective genes of the hormone or certain control gene. So this is possibility of the Rdna technology, whereby you modify the plant which gives the better production with less amount of time, with less amount of nutrient uptake. The Rdna technology is useful for producing a product with some novel characteristic. The best example is the golden rice. In ordinary rice, the beta carotene is not found. It is not found or below limit of the quantification. But the scientist has introduced the gene of beta carotene into the rice, and now rice is able to produce 1.96 to 7.31 ppm of the beta carotene. Then they appeared in a golden form. So this is the way whereby you can increase some nutritional quality of the plant. Seed protein quality can be modified by adding some gene into the seed. Best example is the soya milk. So soya milk is the milk obtained from the soya bean. The gene, as you see in the diagram, is introduced into the soybean. Then soybean are cultivated with this extragenous protein, and from that one the milk is recovered and formulated and prepared. So this milk is a unsweetened, means there is no sugar inside one, and helpful for the patient suffering from the diabetes. Similarly, the seed protein quality can be increased for the oil seeds, where the maximum amount of oil can be recovered. So for the better production of oil droplets in the seed, the genes can be modified. Development of slow ripening gene. Here, the plants and fruits and vegetables are such a modified that they their ripening time can be increased. For an example, the tomato which are cultivated in Punjab, if they are transported to Maharashtra or Bombay, it take at least 3-4 days. So during this period of the transfer, they may ruptured, damaged, or over ripened, and there is a loss of this vegetable and the fruit. So scientists had identified the gene responsible for the ripening. They delete or modify the control system by Rdna technology, whereby such plants can not ripen within short period of time. So increase, or we can say delayed fruit ripening, is helpful during transportation, that they maintain their quality. Similarly, the fruits and vegetables, the skin is very thin. So, due to thin skin, they also damage during handling and transportation. So, some genes are modified which increase the thickness of the outer skin and make the fruits very tough to damage. So this way the plants and agriculture failed. The modification of fruits and vegetables by changing their ripening time and by changing their skin is advantageous nowadays. The plant and agriculture field, the transgenic plant as a source of biopharmaceutics. It is possible to make the edible vaccine. Edible vaccine means the vaccine which is introduced in our body by injection. But scientists finding out the option, the gene of any foreign organism which is an antigen, that antigen gene introduced into the plant that grow along with the plant and in the fruits. So whenever the fruit is given to any child, the child receive the antigen and get that antigen in their body, and along with the eating this fruit, the baby may be immunized, because the fruit contain the foreign DNA, and that DNA act as an antigen in the patient or baby, and against that antigen the antibody will produce, means the vaccine properly work in that body. So now this concept is arose, and research is ongoing, and successfully hepatitis B surface vaccine can be produced by this technology. So edible vaccine is a future concern, and it is useful for the field of agriculture plus medical field. Plant can be converted as a pharmaceutical factories. If you want to produce a hybrid these antibodies through hybridoma technology, it costs about $1,000 per gram. When you use the transgenic animal for the antibody production, the cost is about $100 per gram antibodies. But when you use the same plant for development of monoclonal antibodies, so this transgenic plant cost about $10 per gram. So plant are growing naturally. They do not require some stringent requirements. So when they grow under controlled condition in greenhouse, they can produce the foreign protein. So wash the plant, then identify the protein after harvesting the plant leaf and purify that protein. So that protein can be used as therapeutic protein, enzyme, and diagnostic protein. So this way the Rdna technology is helpful to create such plants which produce your desired therapeutic protein or therapeutic enzyme and diagnostic. The advantage of such plant is that they they the product which do not require the refrigeration, so the product can sustain at ambient temperature. Therapeutic product which can be produced by the plant include the antibodies, enzymes, replacement protein, immunosystem stimulator, immunosystem suppressant, biopolymers, adhesive protein. Okay? The production of immunoglobulin fragment and their assembly in plants that is reported in the tobacco, recently known as the plantibodies. So, immunochromatography or medical therapy is useful for the recovery of plantibodies. Seed based plant bioreactor. An example is the successful expression of human lysozyme enzyme alpha L iduronidase in Arabidopsis thaliana seed. So this is one plant in which the lysozyme enzyme can be produced. So this way the plants now become the factory for the production of human protein or animal protein. The plants are also modified through Rdna technology for the production of primary as well as secondary product, in which similarly the foreign gene is introduced into the plant able to grow this foreign protein, and you can recover from the plant. The best examples are listed over here. Alpha and beta hemoglobin, it is a blood substitute, it is produced through tobacco plant. Serum human albumin, a blood substitute produced from the potato. Alpha trichonxanthin for HIV therapy from tobacco. Alpha interferon, viral protection anti-cancer agent, it is produced in the rice. Hirudin, it is anticoagulant produced from the canola. Protein C, anticoagulant produced from the tobacco. Glutamate decarboxylase for the treatment of diabetes, it is produced in tobacco. Human somatotropin, it is for hypopituitary dwarfism treatment, it is produced in tobacco. Calcitonin, which is a vitamin D stimulator, it is for the treatment of osteoporosis, parathyroid gland carcinoma, and it is produced from the potato. So very good examples are listed over here. They are the therapeutic proteins produced in different plant host system. So this is all about the plant agriculture field, where the Rdna technology is the useful in variety of the way. And in the last I saw certain genes, the genes of thioester, it is for the high laureate oil wheat production. So the table showing you the variety of genes and which company possess the rights to develop the product. So thank you very much for attending the lecture. Next time we will see the application of medical and pharmaceutical